Okay, hello everyone, and welcome to Essen BioScience's webinar series. Thanks for joining us today. Today's topic will be simplifying 3D tumor spheroid growth and shrinkage assays using live content imaging. Our presentation is being co-presented by Kalpana Patel, Senior Research Scientist at Essen BioScience, and she's joined today by Mark Rothenberg, Applications Manager at Corning Incorporated. So we're going to go ahead and start things off today with Mark Rothenberg from Corning, who will be discussing ultra-low attachment plates as they relate to our 3D spheroid assay and other applications. So go ahead and take it away, Mark.
The first slide goes over an introduction to the ULA surfaces used with our microplates. ULA is an interesting surface. It's an inert hydrogel surface that covers plastic to prevent cell attachment. Now, this surface was developed for attachment-dependent cells to prevent attachment to plastic in order to provide a more in vivo-like environment. Now, normal plastic, tissue culture-treated plastic, provides an attachment surfaces where attachment-dependent cells will attach to and stick. And research over the past 20 years has shown that cells that attach to plastic have especially attachment-dependent, have a different physiologic response than those kept in more of a suspension environment. So if you look on the right side of the screen for the ULA surface, you can see how on the top you'll see self-attachment to the surface. And then if you see the hydrogel, cells will basically bounce off the hydrogel and maintain in solution. That is the whole purpose for this product and for today's talk in hopes of producing tumor spheroids. When tumors or basically cancer cells are attachment-dependent cells, but when kept in suspension, they will form a tumor-like or spheroid-like formation. If you go to the next slide, please. The next slide shows two products, and in today's talk, using the imaging, the IncuSite imager, they use the product on the left, the 707 clear 96-well ULA-coated plate. Now you notice by the formation of this plate, these are round bottom plates, which allows formation of tumor spheroids in a unique manner in a single spheroid per well. So what's great about this 707 plate, in terms of it's a low-cost, easy product to use to make tumor spheroids in a screening format, compared to other technologies where there's a lot more preparation, a lot more handling time. Here, this is basically plug and play. The plate on the right is the next generation of the 707 plate. It's a black plate for round bottom tumor spheroid, has a slightly different conformation on the round bottom that allows for quicker tumor spheroid formation, but both plates allow for some really interesting applications in terms of not only tumor spheroid production, but embryonic body formation as well. Some interesting applications that can be used and have been used and are currently published on using these plates. As I said earlier, the embryonic stem cell body formation. You could do cell lineage determination and screening. Another really interesting application, this one that I saw recently at several conferences, is confrontation cultures, where you can take embryonic bodies, mix them with tumor spheroids, and look how they merge together and physiologically interact. And finally, you could do tumor spheroid production. You can do tumor spheroids, you can do screening, a whole bunch of different interesting assays. If you look on the right side of your screen on the top, as I said earlier, cells that attach to plastic and don't remain in suspension have been shown to have a different physiology than those in a tumor spheroid. One easy assay as a proof of principle is you could look at certain tumors produce VEGF production. And you can see here tumors that were secreted VEGF in spheroid formation had statistically higher VEGF production than those just grown on plastic TCT. On the bottom, it's an interesting assay where they actually looked at formed tumor spheroids and then measured inhibition of those tumor spheroids with a particular drug at higher concentrations. All of these can be done using either one of these plates using imaging such as the IncuSight imaging technology for looking at tumor spheroid biology, eventually if you ever want to do embryonic body formation as well. Thank you.
Okay, thanks, Mark. We appreciate you joining us today. And now I'd like to turn things over to Kalpana Patel, who will be presenting our kinetic 3D spheroid assay.
So thank you, Mark and Jason. Before I start sharing with you our 3D tumor spheroid growth and shrinkage assay, I'd just like to spend a couple of minutes introducing you to the concept of live content imaging. So at Essen, we have defined live content imaging as the acquisition, analysis and quantification of live images from cells that remain unperturbed by the detection method, thereby allowing you to perform repeat measurements over long periods of time, ranging from days through to weeks. We believe that kinetics provides a better understanding of cell behavior within a system, and coupled with detailed morphological characterizations will allow you to evaluate the temporal effects of any treatments. So this slide here just briefly describes the key features that enable live content imaging, namely the ability to image live cells, the ability to generate high and rich content data, coupled with key features of live microscopy, and all brought together with a very simple user interface. So we believe that our instrument, called the IncuSight Zoom, really ticks all of those boxes. It's a fully automated two-colour phase contrast and fluorescence microscope, which resides within your standard tissue culture incubator. It has the ability to image cells kinetically, and it has advanced user-trained image processing and quantification capabilities, as well as integrated analysis modules. So I'm now going to spend the remainder of this presentation focusing on our 3D spheroid assay. I think we're very much aware of how 3D cultures and microtissue systems are becoming of increasing interest to the wider scientific community. This is because they're considered to be more reflective of the microcellular environment of the body, for example, in terms of their nutrient and oxygen gradient and their cellular heterogeneity. More specifically, in the field of oncology, 3D cultures are key models, with multicellular spheroids becoming the most commonly studied form. I think if you were to scan the literature, you'd come across a huge range of methodologies that allow you to generate and quantify 3D spheroids. But I would argue that there are potential question marks around their significant relevance, their ease of use, their cost, and their robustness. So to address this, our goal at Essen was to use the IncuSight Zoom to build relevant kinetic tumor spheroid growth and shrinkage assays that are technically simple, robust, inexpensive, very much making them similar to the way we currently run 2D assays. So with that in mind, we adopted one of the more simple and inexpensive ways of generating spheroids. That is, using Corning ultra-low attachment plates that you've just heard described by Mark. This protocol is described in detail and developed by the group Vinci et al. The protocol I'm going to describe to you is a slight modification of that. In brief, cells are seeded into all wells of a 96-well ULA plate. Following a small and short centrifugation step, your ULA plate is positioned within the IncuSight Zoom, where you're able to monitor spheroid formation over 72 to 96 hours. Once your spheroid has reached the appropriate size, you simply remove the plate from the IncuSight and add your required samples. You then return the plate to the IncuSight to continue to monitor and quantify spheroid parameters over time. You have the option of removing your plate and refeeding and refreshing your spheroids every 96 hours, simply by removing some of the culture media and replacing with the exact volume of treatment. This protocol, being very simple and straightforward, is also amenable to both manual and automated liquid handling systems. So this slide here just shows you how successful we were at generating spheroids in ULA plates. Here we have some IncuSight Zoom HD phase images of spheroid created from a range of tumour cell types. You will see that they show different morphologies, ranging from tight spheroids, for example, that achieved with A172 cells, right through to loose aggregates as depicted by MDA cells. The 3D morphology of loose aggregates can be further improved by the inclusion of an extracellular membrane, for example matrigel, at the point of spheroid initiation. You will note that there is a single centrally formed spheroid in each well, and this is due to the nature of the round-bottomed well and the hydrogel coating, which prevents cell attachment. I think you'd agree that the IncuSight autofocusing algorithm works very effectively, and you can see very clear phase images of the spheroids. However, I would say that automated phase-only image analysis is somewhat challenging, and this is currently a work in progress at Essen. For a non-integrated approach, you have the ability to export these images into external packages, such as ImageJ, to allow for further quantification of phase images. So moving forward, we focused on using fluorescently labelled cells to negate the issues associated with phase quantification. So this slide just summarises some of the fluorescent reagents available to you from Essen BioSciences. We have a range of reagents termed Cytolite and Nuclite, red and green. These will allow you to get started with this methodology straight away. They include a range of lentiviral-based vectors, which have been designed to efficiently infect a range of cell types, giving homogeneous expression of either cytoplasmically restricted or nuclear restricted RFP or GFP. Of course, we already also have a range of cell types ready and available for use. You're able to achieve comparable quantification using either of the cell expression of either cytoplasmic or nuclear restricted expression of the fluorescent protein. Both red and green reagents are compatible for use on the IncuSight Zoom, while only the green reagents are suitable for use on the IncuSight FLR. Of course, you can use your own fluorescently labelled cells or vectors. A couple of things to bear in mind is that your fluorophore excitation and emission spectra should be compatible for use within the IncuSight Zoom. And that homogeneous expression of the fluorescent protein is really conducive to high data quality. So I'm just going to move on and show you a video generated on the IncuSight Zoom showing the formation and growth of a spheroid. So here we have a spheroid formed using human lung carcinoma cells expressing nucleus-restricted RFP. You can see that cells aggregate and form into a spheroid over the first 72 hours post-plating. And the IncuSight continues to monitor the growth of spheroid, in this case up to 10 days. I'm now going to show you a second video which will illustrate the image quality attained using the Phase HD channel and the red fluorescent channel of the IncuSight. So what we have here are again time-lapse videos. On the top you can see a vehicle-treated spheroid and a spheroid treated with 1 micromolar staurosporin. I hope you can see that the spheroid is increasing in size and getting brighter for the vehicle control, while we're seeing a decrease in size and brightness of the spheroid treated with 1 micromolar staurosporin. On the bottom, what you can see with the vehicle control and the staurosporin treated spheroid is the ability of the IncuSite to accurately mask all of those fluorescent images at every time point during the duration of that experiment. So we can quantify spheroid growth and shrinkage kinetically using the IncuSite size metric, and these include fluorescence area and fluorescence confluence, and they require you to mask your fluorescent spheroid. The area metric, as I said, is a real-time measured parameter, and it can be used to derive mathematically a volume parameter for your spheroid. This extrapolation to a volume parameter assumes that your structure is spherical. Currently, the area metric is expressed as fluorescence confluence, where area expressed in microns squared is a function of the field of view. The area metric reported in microns squared will be available as part of the imminent free software upgrade available to current Zoom users. The fluorescence confluence metric is a suitable alternative for all FLR users and current Zoom users. So I've described to you how simple and straightforward it is to create spheroids using ULA plates, and for you to image them and track their size using the IncuSite Zoom. I'm now going to demonstrate to you how simple it is for you to control the size of your spheroid by simply controlling the number of cells seeded per well. This may be of importance to you, for example, if you wanted to model key features of tumours, where it's been reported that spheroids with diameters in excess of 400 to 500 microns typically have a central necrotic core surrounded by a layer of quiescent cells and again by a second layer of proliferative cells. While I'm not showing you the data here, we have gone on to demonstrate spheroid size is homogeneous within a plate, between plates, and across multiple experiments. The plots below the images of area versus seeding density and volume versus seeding density further go to validate these size metrics, in that you will see both area and volume increase proportionally with the number of cells seeded per well. IncuSite Zoom has the ability to automatically generate kinetic plate view visualizations, and here is such an example. What we're looking at here is a plate view from a 96-well plate assay looking at the effects of 3 compounds on proliferation of A549 nut-like red spheroids. What we can see from this time course that it's very easy to determine the effective inhibitory concentration of the treatments, and also the time course is for those compounds, and the consistency and reproducibility of replicates within the test groups. That data can be automatically exported and used to generate time course plots as shown in the top of this slide. Area under curve analysis for the whole of that time course can be used to generate concentration response curves for your treatments and go on to derive pharmacological data, in this case PIC50 values. The dashed line on the concentration response curves indicate no growth of the spheroid. So you'll see here staurosporin causes a spheroid to shrink to a size smaller than it was at the start of the experiment. And this is not observed with the cytostatic effects of cyclohexamide. Now, you can also continually validate your quantification through visualization of images from your IncuSite. And here we have such images taken from the experiment I've just described. On the top, we have the vehicle control spheroid, images taken from 96 right up to 258 hours, And what you can see, which is confirmed by those plots I showed you previously, an increase in spheroid size, and to note the spheroid is also getting brighter. The effects of 1 micromole withdrawing results in the shrinkage of the spheroid and a dimming of its brightness. Now, interestingly, we've come across compounds which cause a time-dependent reduction in spheroid fluorescence. And this is exemplified with the effects of 10 micromolar mitomycin on the A549 nuclite red spheroid, where from 96 hours and beyond, in the presence of the compound, we see marked reduction in spheroid fluorescence, but do not see a change in the size of the spheroid. This is clearly not due to the instability of the RFE, because this observation is clearly not seen within the control group. So, if we were just to look at that data in more detail, for example, using the data attained at 198 hours, and if we were to quantify the size of the spheroid using phase confluence, we would see that the spheroid treated with both 0.1 and 10 micromolar mycin returns equal size metrics. However, if we were to consider quantifying the spheroid based on the spheroid intensity, we would see that the intensity reduces beyond a reduction in size. So just to elaborate on the fluorescence intensity metric in a little bit more detail, as I said, this metric is based on the brightness of the spheroid. It is the mean fluorescence for the entire image, and for it to be successful and truly reflect the intensity of the spheroid, it assumes negligible contribution from background fluorescence. And this is the case when using Essen nuclite red or nuclite green reagents. This metric, which we've termed mean image fluorescence, is available very simply as a scan metric from your IncuSite and does not require any masking of the image or the creation of image collections or a processing definition. And we believe that it will go some way to providing you with an index of 3D morphology or the cell health. Taking all of these features into account, we recommend this metric and prefer it as a metric for quantifying spheroid growth and shrinkage. So, while not a direct measure of size, the mean image fluorescence metric is a useful surrogate. And just to validate that further, what you can see here is that we see a very nice and good correlation between the mean image fluorescence of the spheroid versus the size metric here in area. So just returning to another 96-well plate view visualization, which is exactly the same as what I showed you previously, but here looking at the time course of mean image fluorescence. And again, I hope you can see that we're still able to achieve very consistent and reproducible data using this metric. So I just want to introduce you to some preliminary data that we've achieved using the 384-well Corning ultra-low attachment plate that you heard Mark touch on earlier. So what we did here is use the 96-well protocol that we developed without any modifications or optimizations, and just substituted the 96-well plate for the 384-well plate. And what you can see is that the IncuSite was very successful in being able to image spheroids from that plate type. And looking more closely at some time courses where we're tracking mean image fluorescence versus time for spheroid cells seeded at different densities, you can see that the metrics track each other reasonably well across the two plate densities. So I've described to you and summarized again how simple it is to generate 3D spheroids and quantify them using the metrics for measuring spheroid growth and spheroid shrinkage within the incucyte zoom. And this has really enabled us to perform 2D and 3D proliferation assays in parallel with somewhat ease, whereby we can use the same incubator, the same compound plate, same cells, etc. And this is exactly what was done here. You can see time courses from a 2D assay and a 3D assay on the right. So truly enabling us to perform some comparative and quantitative pharmacology. Now, while similar observations are observed with the two formats, 2D and 3D, what you'll note is that the duration of the experiments was very different. So while the 2D completion within 2 days, the 3D experiment had to be run out for 10 days. What we have here are some images taken from a parallel 2D assay and 3D spheroid assay, looking at the effects of cyclohexamide on A549 nucleate red cells over a 90-hour time period. And what we can see on the top here are images from the 2D assay, a vehicle control well, where cells proliferate over the 90 hours, approximately an increase, 8-fold increase in confluence. If you compare to what's happening to the vehicle control spheroid, you can see that there's a very small fold increase in size, approximately 1.3-fold. Cyclohexamide at approximately 1 micromolar causes inhibition of proliferation in 2D. However, the effects in 3-fold in the 3D spheroid assay, you'll see, cause very little, if any, inhibition of proliferation. So just capturing that full time course across the whole concentration ranges tested for cyclohexamide, I think you'll see that there's a marked difference in the temporal profile of cyclohexamide-induced inhibition of growth, with cyclohexamide being approximately 10-fold more potent in the 2D assay versus the 3D assay. The significance or relevance of this at present is not understood. However, to address this further, we are profiling a range of compounds with different mechanisms of action in the same assay. So finally, I would just like to summarise by saying, using the Corning ultra-low attachment plates, we're able to generate a technically simple and robust and economical way to make 3D spheroids in this plate type. And this protocol is almost as simple and equivalent to running your 2D experiments. Using fluorescently labelled cells within the incucyte zoom, we've been able to build, fully validate a kinetic spheroid growth and shrinkage assay. The incucyte metrics, whether they be size-based or using the mean image fluorescence intensity metric, are very informative and can be gathered in real time to monitor spheroid growth and shrinkage. We believe that spheroid fluorescence intensity measurements, which do not require any masking, may provide a useful index for the 3D nature or cell health of your spheroid. The images and time-lapse movies provide very important information and help validate spheroid morphology and the effects of test samples on them. So in summary, these assays should prove very useful for medium throughput quantitative pharmacology for observing the effects of your test samples on the growth and shrinkage of spheroids and allow you to run parallel experiments within 2D and 3D systems. So with that, I'd just like to conclude my presentation. But before I do, I'd like to introduce you to a colleague of mine, Dan Appledorn, who is our Application Support Manager, who will also be online to help address any questions you may have. Thank you.
Want to remind you that you can type any questions you have for either Kalpana or, as she mentioned, we're joined by Dan Appledorn, who's our R&D Applications Manager here at Essen, or certainly any questions for Mark with regard to the Corning ULA plates. You can type any questions you have in the chat box in the lower left-hand corner of your screen, and we'll go ahead and address as many of those as we can.
Thanks. Yeah, I see a number of questions here. The first question here is one addressing the utility of this assay in an IncuCyte FLR or an IncuCyte Zoom. The question specifically is it possible to use the IncuCyte FLR to do the spheroid assay using GFP-labeled cells, or is the IncuCyte Zoom the only option? As Kalpana mentioned, we believe that this assay can be done both in the IncuCyte FLR and in the IncuCyte Zoom. In the IncuCyte FLR, certainly the GFP fluorescent marker would be the only option, whereas in the IncuCyte Zoom, we would recommend that you use the red channel to image and quantify spheroid size. For those users out there who are familiar with the IncuCyte, you'll know that or in fluorescence microscopy in general, you'll know that the background fluorescence contribution from the media can be have a significant effect on fluorescence intensity. So if you were doing this assay in the IncuCyte FLR, we would recommend that you mask your spheroids in the green fluorescence channel as opposed to using the image mean fluorescence as Kalpana has talked about. Either way, you should be able to successfully achieve quantifiable data for this assay in either instrument. The next question, you mentioned using extracellular matrix material such as Matrigel in the spheroids. Can you expand on this? Sure. So we have found, and this has been shown in many different publications, and our data suggests similar results, that adding a small amount of specifically Matrigel to the growth media can result in different spheroid phenotypes. So a spheroid that forms a loose aggregate, cell types like MDA MB231 cells can form a much tighter spheroid if you add a very small amount of Matrigel, and that's on the order of 1 to 2% of Matrigel to the growth medium. So we find that, like other protocols, adding a small amount of extracellular matrix can change the phenotype of the spheroid. The next question is, do cells have to be fluorescently labeled to measure growth kinetics, or can you do this in phase contrast? Our current implementation of this assay on the IncuCyte does require cells to be fluorescently labeled. Many of you who have used the IncuCyte before know that we can process images in the phase contrast channel. And we can do that using these spheroids, but we find that oftentimes the mask that is generated doesn't perfectly fit the spheroid. We'll catch scratches on the bottom of the plate, or we'll lose some of the contrast of the spheroid in the middle. And so you're certainly welcome to try using the phase contrast channel to measure spheroid size, but the fluorescence channel is much more reliable and therefore is recommended for this assay. As Kalpana mentioned, if phase is of particular interest, there are other third-party programs that you can export the images to in order to quantify on the phase channel. Having said that, we also know that there are many advantages to using the fluorescence channel. For example, many of our current customers are already using this spheroid assay to measure and quantify spheroids in co-cultures using a stromal cell and a mixture of stromal cells and tumor cells. The stromal cells labeled one color, the tumor cells labeled another color, and looking at the interaction of those two cell types in the formation of the spheroid. And that is achievable only if you have fluorescently labeled cells to differentiate between one cell type and another. Next question is, not all cells form spheroids. Can this assay be used for cells in a way to look at cells that do not form spheroids? Certainly, because we are not providing a definition for the round bottom plate, you can look at suspension cells or cells that don't form spheroids in those round bottom plates. Next question here, is the centrifugation step essential for spheroid formation? What happens if you don't spin? That's a good question. We have tried with and without spin, and we have many experiments that if you don't spin, that the spheroids form one nice homogeneous spheroids in the middle of the well, and then just a few experiments where if we don't spin, that we see multiple spheroids form in the well as opposed to one single spheroid, which can change your results a little bit. So we have gone with the recommendation of the spin step. We think that this is cell type dependent in many ways. With one cell type, we'll see multiple spheroid formation without the spin, and other cell types we don't. So it's just a general recommendation, but of course, with any protocol, you're welcome to modify as you find necessary. Next question is, how many cell types can produce 3D spheroids on ULA plates? That's a good question. We have tried several, maybe half a dozen to a dozen cell types on these plates, and they seem to form spheroids, but these are also, we pick and choose cell types that we know are likely to form spheroids on the ULA plate. So our experience right now is limited. We're certainly interested in hearing your experiences with your cell types as well. So that's going to end our Q&A session here. That's all the questions I have in front of me. If you have additional questions, as Jason has mentioned, please feel free to contact us in bio. If I haven't addressed a question, there are a few on here that I haven't addressed. If I haven't addressed your question, I will be sure to follow up with you on a personal basis to address your question over email.
Okay, at this time, we're going to go ahead and conclude our presentation for today. We thank you very much for joining us. And special thanks to Mark Rothenberg from Corning for joining us as well and answering questions about the ULA plates. So we appreciate Corning's participation in this as well. If you have any questions on the 3D spheroid assay, feel free to go ahead and email us. Again, that's sales@essenbio.com. We encourage you to check out our website as well, which has information on this and all of our cell player assays, as well as the InQSight Zoom live content imaging system. And that web address is up on your screen now, essenbioscience.com. And be sure when you're there to connect with us via LinkedIn, Twitter, and Facebook. We often post exclusive content on there and invitations and things like that that you can't find anywhere else. So again, we thank you very much for joining us today, and we'll look forward to seeing you next time.